Hello, everyone. Well, first, I'd like to thank the organizer for giving me the opportunity to convey some excitement about the RNA modifications. So, okay, so again, I'm going to talk about epitranscriptome, the word commonly used today about RNA modifications. And this, some of the work in my lab was funded by NRGRI. We have a Center of Excellent Genomic Sciences. And we also have an R01 for the meeting tomorrow, but actually that one is not on RNA modifications, but nevertheless, you will hear more about it tomorrow. Okay, so we all know that since we're talking about molecular biology, of course, then we're going to focus on RNA. And in fact, the epitranscriptome here, it's sometimes I call it the RNA equivalent epigenome. EP, right, stands for something beyond. So epitranscriptome then is something more than the transcriptome. So the way I normally think about this is that here, by the way, there's many RNA modifications being discovered in biology in the three kingdoms of life. And in humans, if we take our RNA from our cells, I would say that our RNA is first born with the 4 letters, but then after the chemical modifications become over 50 letters. So there are over 50 chemical modification types that's been found so far in human biology. And of course, many of these modifications have simple chemistry-wise, from simple methylation, that would be the simplest, or base isomerization, such as pseudouridine. But also it can be as complex as something like building an X-ray ring on top and so on, and something else get added to that. And sometimes these modifications are catalyzed, of course, by genome-encoded enzymes, and these enzymes range. So to make something like this actually requires 6 steps of chemistry with 6 group of enzymes. So you will get a lot of, I would say, overall. In the human genome, we're probably looking at at least over 100 proteins or genes, protein coding genes that are actually involved in doing RNA modification alone. Okay, so here just zoom into a little more detail. Our work deals with mammalian systems, but also microbiome. But so I normally talk more about mammals. So there, as I mentioned before, 150 types. These are all about 3 kingdoms of life: bacteria, archaea, and eukaryotes. But here in some of these modifications in the mammalian systems, they appear in different type of RNAs, right? So we're familiar with ribosomal RNA, tRNA, nuclear RNA, basically spliceosomal RNA, and messenger RNA. And so as indicated here, so not all the modifications appear in every RNA family, and to our tRNA turned out to be the most, contains the most elaborate set of modifications. Again, amount of 50 chemical types in a human cell, about 40 of them actually show up in tRNA. Okay, so so the RNA modification got a lot more attention today for this very simple reason. That's I assume that everyone in this room have received at least an mRNA vaccine. The mRNA vaccines is actually made not out of the 4 standard nucleotides, rather all the uridines in these vaccines. This is are substituted with a modified nucleotide called N1-methyl pseudouridine. And this is the actual sequence of the Pfizer-BioNTech vaccine. And just to expand over there is that just to show you that every uridine residue is actually modified. But this N1 pseudouridine here doesn't occur. It is found naturally a lot in archaea. Not really, but so far it's only appearing as some kind of an intermediary form of a different type of RNA modification in humans. So the really naturally occurring modification, the most abundant in all 3 kingdoms of life, is this modification called pseudouridine. So what this is the base isomerization. So it's just illustrated as a chemical example. Uridine looks like this, but if we take the base off, or the enzymes do that, of course, take the base off, and then connecting this carbon here with this carbon 5 over there, and now we end up with something called a pseudouridine. And this is if you just take total cellular RNA, digest everything down to mononucleotide, and do a mass spec analysis, you will find out that actually pseudouridine has the highest content because it appears is present in a lot of that in ribosomal RNA and tRNA, and a little bit some of that in the messenger RNA too. Okay, so where are the modifications? So here's a structure of the human ADS ribosome. we have approximately 100 two-parallel methylations and sites and 100 pseudouridine sites and about 10 base modifications. And these modifications this is just how they're mapped in a 3D structure. And they're required for ribosome maturation. And also they, of course, you need the presence of most of them, not all of them, but most of them at the same time so that the cells can actually make a good mature ribosome and make synthesized proteins. And these modifications, turn out they're dynamic, the way we call it, because even though it says here about 210 modification sites, if you really look at the human and different tissue cell types and so on and so forth, or different cell states, if we using a more sophisticated mapping technologies, we can find, in fact, the modification, the stoichiometry at each of these sites, or as many of these sites, can vary. So this is kind of the cells really don't make a simple, just one single ribosome with these modifications. Rather, they make many different ribosomes. They differ somehow in their, the modification status. Okay, then tRNAs, these are this is a sort of composite diagram made by a graduate student of mine when we wrote a review here. So these were just the chemical types of tRNA modifications located in different locations. Again, anticodon loops or stem loops over here. And in terms of chemical diversity, the anticodon loop region has the most modification types. And because these are the ones that exactly has to read the codons. And so many of these modifications are really used to fine tune how the specific codons are read under different cellular conditions or different cell types and so on. And also it minimizes frame shifting, but then we also have many modifications outside of the anticodon loop. Those are sort of not as well studied as the ones in the anticodon loop, but nevertheless, we can make some kind of a bullet point here to say, oh yeah, they're required for stability, folding, ribosome binding, localization, and so on. Okay, then for messenger RNAs, and this is just, again, coming from the review I mentioned before. So we have approximately 10 or so chemical types that's been found as are present, and shown in the boxes here are just the two most abundant ones. So these percentage here, what that means is the most abundant modification is called M6A or N6-methyladenosine. It's a methylation form here. And this is present at approximately 0.5% of all the A's. That means every 200 A residues, you find about one of those. And if you look at taking the average size of the human mature messenger RNA, we get approximately 3 M6A residues per messenger RNA molecule. And then the second most abundant one is this is our old friend pseudouridine. Okay, this is sort of the excitement started about a decade ago now, over a decade ago now, that it's the discovery of how, in fact, human cells can use, and the M6A can not only be installed, but can also be erased. And this is how we sort of start borrowing, the field start borrowing terms from the epigenetics, where if you to do the biology of RNA modification, just normally we will need a group of proteins, we call them writers, that put a modification in. And then for some modifications, only so far for some modifications, we find enzymes that are erasers that can actually take the modification out if needed. But really, in order for cells to do something with them, at least for mRNAs, we will need another group of proteins. They're called readers. And the readers are the ones that so the writers and erasers are the ones that work together to establish as kind of a modification pattern in the transcriptome. But then the readers are the ones that pick out a specific mRNAs to be modified. And then depending on which reader it is, and then these subset of mRNAs will go through a lot of the processes that are required to be done for the messenger RNAs. Again, this is just sort of the catalytic subunit, the gene names for in the mammalian cells. So for instance, the writer for M6A, there's the gene called METTL3 or metal L3, but in reality, this writer is really is part of metal L3 is part of a big complex that does a lot of the regulation, and then this complex has, I think, 7 or 8 proteins or so. And then we have 2 erasers been discovered, and one is called FTO, and the other is the RFBH5, and those are the ones, again, as catalytic subunit. They are also form protein complexes that that can do many things. And reader proteins, there are so far many you can discover, but these are just sort of the protein families. They call YTHs, so the proteins with YTH domain. They are M6A readers. We have proteins that are called HNRNPs. Those are stand for heteronuclear ribonuclear protein particles. Those are the ones that code messenger RNAs. They're very abundant nuclear proteins in generally that code messenger RNAs, and then they regulate mRNA splicing stability and export. And then there's also another group of protein, family of proteins called IGF-2BPs. Those are the ones that generally sit in cytoplasm that actually guide the mRNA to do something interesting. Okay, so how do we study modifications here? Of course, we got to do sequencing, and to get the site-specific information at the transcriptome level. So these days we do sequencing. We have next-gen NGS, or we have the third-gen TGS. So next-gen is simply typically Illumina sequencing is still used widely, mostly today, and then I think to this room of people, but at this point, I don't need to explain how Illumina sequencing works. But the TGS here, again, these are the PacBio and nanopore sequencing and so on. And the PacBio you will still have to convert the RNA into cDNA because PacBio doesn't have a commercial platform yet to directly sequence RNA, but the nanopore does have the commercial platform. You can do a direct RNA sequencing with nanopore, and this is how we get our modification information. So exactly what do we actually look for? If we do like NGS or TGS sequencing, what are the actual data we look at to say, oh, there's a modification, we call it. Okay, so this is how, in NGS, this is how we call it. We measure modifications by a thing which we call mutation and or deletion. So what this means, here's some examples that show you. So here's some real sequencing data. And we have this modification here in a specific tRNA here, right? tRNA GCU. And it has a methylation. And as you can see, this methylation here is on the Watson Crick phase. So when we use the right type of RTs under the right conditions, when our reverse transcriptase sees this modified nucleotide, it doesn't just stop there. Rather, it somehow just muddles through. Okay, so when it goes through, then it doesn't just put a G back there. It just can't. So instead, what it does is that you put a bunch of something else in, right? And A, C's, and T's, and just everything completely depends on the modification type and the sequence context. So now, if we take our sequencing reads, we map it to a reference sequence. In this case, tRNA GCU, we start with a C. But then if we map it, if you look at this position, it turned out that, wow, 70% of those reads that map to that position, actually, they're not a C anymore. So this is, we call it a mutation. A deletion is something like this. So this is just another example. There's another modification called Q-usine. This is the chemical structure. If we do a chemical reaction with this opens up this ring, then now, so if we don't do the chemical reaction, we will have deletion. Just means, again, when we do our sequencing mapping to a reference sequence of this tRNA histidine here, deletion is all of a sudden a nucleotide or one or two are skipped. And then, if we don't do this chemical treatment, this is the black line here, we get no signature. But if we do the right chemistry and then build our sequencing library, we'll get actually, in this specific case, a double nucleotide deletion that indicating that in fact Q-usine is present at this position 34. And finally, you can do other fancy things like this modification here with a SIO group here. Now, this one, if you do a chemical treatment, we get both, only with, in the presence of the treatment, we get both mutation signatures and deletion signatures. So this is how you can do the same thing for to map messenger RNAs this way too. Okay, what about nanopore sequencing? So this is one that at least is one of the approaches that commonly people use, and this is one we use in my lab to map pseudouridine modifications, and so this is, in this case, is measure modifications by machine learning or training data. Nanopore here, simply right here we have a protein pore with a current going through, and here's ideal data, of course, but real data looks like something like this. So in order to really study modifications, we do a direct RNA sequencing, but then what we really need is the training data for pseudouridine using a sample with known pseudouridine size and stoichiometry. So again, this is absolutely crucial, of course, to really get good results. But once we have this, these kind of nanopore data, then we can build a machine learning model. And in our case, our machine learning model eventually examines 12 different parameters from the nanopore data, direct RNA sequencing data. So this allows us then to map pseudouridine at the sites with some reasonable confidence. Okay, and again, in this paper, we actually studied a biological problem using this approach, is how does pseudouridine human mRNA transcriptome respond to interferon treatment. Okay, so I want to advertise here that is actually the National Academy, US National Academy of Science and Engineering Medicine, and some just produced this report of charting a future for sequencing RNA modifications. And I'm a committee member of this report, so I guess I get to really tell you how great this report is. And in fact, I mean, it's freely available as a PDF. You can just download online. And this was published, I think, in April 20 this year. And what this report does is that it actually has a very lengthy review of the status of the field and even the history of the RNA modification studies. And of course, it offers a lot of guidelines or at least recommendations for how we gonna do better to study RNA modifications in the future, and so on. And in fact, this is a lengthy list here. If you just skip out this actual history and the status of the field and all that, and eventually get down to this recommendation list where it says, okay, here we hope that as a committee that perhaps there is a course of something more specific in the next 15 years to allow us to really study RNA modifications and in this context of human health and disease and other sets of subcore biology. Okay, I think that's it. Thank you for your attention.
Thank you. That was a great talk. This is less of a question and more of a plug for the NASM report. Yep. For those of you who didn't see, it's about 260 pages. If you're interested in reading it, but you don't really, or you want to learn more about RNA modifications, the NASM does a great job in about 12 to 14 pages of giving a wonderful summary about it. So thank you for your talk. It was wonderful.
Yeah. Okay. Well, thanks. Yeah. Thanks for pointing that out.
Are there take-home messages that we know yet about pseudouridine and M6A in messenger RNA and how their modified transcripts are functionally different from unmodified transcripts?
Right. So you're talking about messenger RNA specifically
RNA,
for pseudouridine, for instance. So M6A, of course, has been studied very extensively. I mean, things I would say 2011, 2012. So there's M6A is basically is mostly the reader protein catches them, and these reader proteins have other domains on them. Simple example, YTH proteins have domains that are, we call them, low complexity. They can go through condensate formation, and they can interact with other proteins or other RNA and so on and so forth. So again, I think the M6A function is whichever reader protein gets the mRNA in the M6A modification dependent manner, and then that goes through the biological process that way. And of course, as you can imagine, it's a messenger RNA, so it's involved anything what a messenger RNA can do. So pseudouridine is still difficult to understand what it does biologically. I think there's some kind of bullet point things. Oh, if you have more pseudouridine, the RNA are more stable. I don't think that, and how that works is actually not clear either. So one important thing here is that do we have pseudouridine readers? That would be one example that we can go through the same question for the M6A. So far, okay, so it eventually gets to the nitty gritty. So what do you mean? What's a reader, right? So it turned out that there's different mechanisms or different chemical mechanisms to molecular mechanisms to read something. You don't have to read it just to see the chemistry change. Rather, you can read it through changes in the RNA structure and so on. So by that definition, many RNA binding proteins can become, which we call readers. Now, pseudouridine has been talked about having many of those things. But how they go through the consequence of those reader protein action of pseudouridine is unclear. For ribosome translation, though, pseudouridine is known to, in the coding sequence, it is known to slow ribosome down. So you would think, oh, slowing down is not good. No, actually, that's not true, because in translation, it's not about how fast you can go. Rather, is you want to put polysomes, multiple ribosomes, at the right space from each other. Just like on a highway, you want to drive a car fast or slower, but it's not as important as that. it's important is that everybody drives same speed, then you can actually get to the close the most efficiently. So the pseudouridine in the mRNA may do something in that way, but that's actually still needs to be proven. Yeah.
So in order to characterize the RNA modifications using cDNA-based sequencing approach, does RT enzyme make a huge difference?
Yeah, the RT enzyme is absolutely crucial. Which one you use, each one can give you a different pattern, right? So that's, yes. I mean, some RT enzymes gives you nothing. Some and it also depends on the modification type. The curiosine thing I showed you before in our publication, we tried 7 RTs, only 1 worked.